Hello everyone, welcome to you all. Today we are going to discuss the phase 1 of the clinical trials and investigational new drug application IND. Clinical trial is one of the four most significant thing which plays a significant role in the drug development. It not only helps in development of new drug molecule but also helps in various directions for the further research. As you know that clinical trial is a systematic study where the laboratory animals and healthy volunteers, further diseased volunteers, are tested with the new chemical entity called pharmacophore. Whereas we see that pharmacological profiling, toxicological profiling, safety, efficacy, quality, pharmacokinetic, dynamic. From every angle, we have to see that a molecule is safe enough for the human use in various target called illness or disease. So, a systematic study of pharmaceutical products are going to be nutraceuticals, cosmeceuticals, pharmaceuticals in the human beings for the effectivity. That is what we do the clinical trial. So it is called as first time in man or first in man, FIM, sorry, studies. So this is a phase where 20 to 100 healthy volunteers are taken up. Their informed consent form is filled up. Informed consent is taken from them to participate voluntarily in this study. So, as per the biomedical ethics and the clinical research pathway, this process work on. The main objective of the phase 1 studies is to establish a safe dose range, biological dose range. And phase 1 trials also evaluate drug metabolism, structure activity relationship, mechanism of action. So, what should be a biological dose which can be administered in a human being has to be finalized. What are the various objectives of phase 1 study? To check, quantify, estimate the safety and tolerability, means how much a bearing capacity determine a human body can take up. Because if we do titrate the dose that is harmful to the other vital organs of the body, for an example, ECM paracetamol, this is the label statutory warning written into it. The higher doses of paracetamol may cause hepatotoxicity. So hepatocytes, liver cells, and furthermore, in terms of safety, if we talk, we have to also see for the drug related problems or medicine related problems, which comes during the concurrent use, like adverse reactions. Means what is the extent of the adverse reactions? What is the range? What is the causality assessment? In how many number of patients this is detected? And coming on to the part like management. The second objective is determination of pharmacokinetics to set the duration of action of the drug, hepatic clearance, dosage form designing, all the dosage form characterization, and also the route. And there is a tendency, as I earlier discussed about adverse drug reaction, coming on to the part of overdose, side effect, adverse effect, or drug interactions. So this leads to determine a pharmacokinetic profile of a molecule. Then comes the assessment of the pharmacodynamics, where we determine the mechanism of action in humans and the structure activity relationship and the early measurement of activity, where we have to determine the potential therapeutic effectiveness. So what is the process? We take 20 to 80 healthy volunteers, and the time duration required is 3 to 6 months. And our focus is on assessment of clinical safety. Whereas in the phase 1, 70% of total lead compounds which are being tested clears or pass endpoint they achieve of their phase. Every phase have their two things. First thing is called as objective, second is at the last end point. If that end point of that phase is achieved, then only the phase promotion takes place. So the drug will go into the phase 2. So at phase 1A, where the 70% of potential drugs passes, and definitely there is a medical clinical pharmacologist, clinical pharmacist, clinical research team consistent of various peoples of professional domain experience. So here also we execute the phase 1 before it that we go for the clinical trial registry, CTRI, clinical trial registry of India, and biomedical ethics and application of IND, and then we start as per the informed consent process. Then the patient should be healthy in the phase 1. I mean to say there should not be any kind of disease, target disease patient. There may be any kind of confusion. So that should be outweighed among the research team also. And a normal healthy volunteer means pharmacological or toxicological approach you want to check in the human being. So the vital organs or the organs function capacity work normally to see of the metabolism and excretion of this unknown product which we are developing or drug we are developing in the process called clinical trials. So we take up the informed consent where who enter in the phase 1 studies, and we have to tell them verbally that this is the process, this is the duration, and this is a registered trial, and this is the clinical experiment. And anytime you feel that to withdraw the study, you can withdraw. So after verbally, then comes on to the written part where their signature is taken up, and after the signature, their left thumb impression has to be taken up. And the after this, expected side effects in the preclinical animal studies are represented, and the possibility that other unpredictable side effects can occur must be stated. So afterwards comes the study. We started very low dose, as I have told in the microdosing phase, 100 by 1 by 100 part, which is toxic to animal. So we take a group of patients with higher dose. So dose escalation takes place, reaching to the maximum tolerated dose and lethal dose, therapeutic dose, called biological dose. So to get the therapeutic index, we do this studies, which is one of the best conclusion for the upcoming phase 2 trials. And usually in the phase 2, which is a therapeutic confirmatory phase, is about 4 by 5th part of the maximum tolerated dose that is 80%. So the phase 1 studies are there to determine an optimum biological dose, OBR, the dose associated with maximum desired biological response for which toxicity is still acceptable. So schedule of drug administration in phase 1 studies is determined from preclinical testing. This is one of the things. Then there are some testing which has to be done, like laboratory biochemistry, hematological, biochemical, or the diagnostic marker, diagnostic tool, diagnostic criteria for the targeted disease for which we are conducting the clinical trial. And in this segmented part, as per the medical lab science, is this performance to monitor treatment effect on the function at major body systems. So blood level of the agent are measured in order to establish the concentration, rate of disappearance of the agent from circulation, and we need to correlate the blood level with the specific side effects. Coming on to the therapeutic outcome or the anticipated benefits, what we see as the part of the therapeutic activity or output, we calculate as the risk and benefit. So if the 10% risk and that is the calculated one, and the 90% anticipated benefits are there, we go with the clinical trial. So in the anticipated benefits, phase 1 studies are primarily directed toward establishing a dose for further testing. There is always the possibility of therapeutic benefit, and this study which is very good process. So our drug development typically consists of biological screening and compound selection, its purification, synthesis, laboratory animal research, animal testing, and permission from the IRBs, and specifically going on to the other parts, which where the investigational new drug is submitted, and also the things which go into the specific part. So the phase 1, phase 2, phase 3, and this usually is one of the thing that at the very primary part you apply for the investigational new drug. So various stakeholders of the clinical drug development process, like you say clinical research associate, coordinator, principal investigator, or the co-investigator, clinical research team, or various people who look after or review this whole clinical trial process called institutional review board, the group of intellectual scientific clinical trial persons who have reviewed previously these interventional trials where the humans are directly exposed to the unknown compound which may causes toxicity and maybe sometime organ failure or loss as per the past history of the drug developments. So, why this checkpoints and this so much of documentation is there? Because this review helps out to cut out any toxicity or anything which may be harmful in the mankind use. So, this is one of the various processes where the IND, NDA, new drug application, then abbreviated new drug application after the audit, or the phase 3, after the phase 3, the internal and the external audits which are conducted. And thereafter the compliance report, a report comes up which we do revise the things, do the performance, and in a triplicate trial master file, and that abbreviated new drug application is ultimately then again submitted, and then there's the marketing authorization, and thereafter then we move towards the phase 4 called as the post marketing surveillance or pharmacovigilance, and from thereafter we also go for the translational research called phase 5. So here after this, in the phase 1 I have completed. Now and second part of this session is IND, investigational new drug application. So the FDA's role in the development of a new drug begins when the drug sponsor has screened the new molecule for pharmacological activity or acute toxicity potential in animals which want to be tested in the diagnostics for the purpose or therapeutic potential. So FDA and their as per Federal Food and Drugs and Cosmetics Acts, and become a new drug, a specific requirement to the drug delivery system. So IND is just a kind of, I would say that information, a note, a summary, which is filed to the regulatory body. Every country have their own regulatory body. If I talk about India, it is Central Drug Standard Control Organization. If I talk about Great Britain, MHRA. If I talk about Australia, Therapeutics and Good Administration. If I talk about Switzerland, Swissmedic. If I talk about Canada, Health Canada, etc. So various nations have their own regulatory authority or regulatory body which they work on this segmented part. So there are three types of IND, investigational new drugs. So first is investigator IND, second is emergency use IND, and the third one is treatment IND. So the investigator IND is something where it is submitted by a physician who both initiates and conducts an investigation. Investigation is something to find out and under whose immediate direction the investigational drug is administered or dispensed. It means the interventional trial, and this is called as also research IND, where we are going to study an approved drug molecule by the regulatory authority, and this product has a new indication in a patient, so that is why. Second type of IND is called as the emergency use INDs, where the FDA authorized to use an experimental drug in an emergency situation, but which does not allow as per the IND in accordance with the 21 Code of Federal Regulations, Section 321.23 or Section 312.34. So it is just like emergency, like endemic or pandemic, where we saw pandemic COVID-19. So likewise, then the third part of the type of the IND is called as the treatment IND, where the submission for the experimental drugs which show the potential for testing serious or immediate life-threatening situations, acute, subacute, or the chronic diseases, illness. And these conditions which are finally clinical work is conducted, and the various regulatory authorities and the auditors called inspectors during the NDA phase after the phase 3 reviews this IND. Thereafter, another kind of classification of IND is there called commercial and non-commercial. Commercial is which permits the sponsors to collect data on clinical safety effectiveness needed for application for marketing authorization form of new drug application called NDA. Whereas the research or the non-commercial permits the sponsors to use drug in research to obtain advanced scientific knowledge of new drugs. So there is no plan to market the product for the research purpose only will serve. So what are the various contents? There are three broad categories: animal pharmacology, manufacturing information, clinical protocol, investigator. So this all the things are filled, and study is approved and reviewed by the IRB, Institutional Review Board, and clearance by the IEC. And as per the new drugs regulations, the IND is submitted, and the sponsor have to wait for 1 month, 30 days before initiating the clinical trial. During this time, the regulatory authorities have the opportunity to review the IND for safety to assure that research subject will not be subjected to any unavoidable risk. Now, what is the format of IND? The cover sheet, as per the form FDA 1571, should contain name, address, telephone of the sponsor, identification of phases, commitment not to begin the clinical trial until IND is approved or registered by CTRI or the approval given by the regulatory authority. Then commitment by IRB, form number 56, commitment for conducting clinical trial with the regulation GCP, good clinical practices. Then the name, title, designation, connect, mail ID of the monitor and the persons, clinical research associate, coordinators, auditors, and the internal principal investigator, and the whole sole team, their I-card, etc. And the signature of the sponsors. Then the table of content, introductory statement, general investigation plan, investigator brochure, study protocol, steps, investigator facilities, and the institutional review board data. Then coming on to the chemistry, so QSR or the functional group, chemistry, manufacturing, quality assurance, quality control data. Then the pharmacokinetics and the pharmacodynamics, so pharmacological data. Then the toxicokinetics, toxidrome, toxicity, doses, variation, so toxicology studies and its data, and the previous human experience, if any kind of experimentation done, their experiences. And for the same, Center for Disease Evaluation and Research, CDE, are having this consultation program also, and there are guidance documents given by the FDA which one can see to design production, manufacturing, testing of regulatory products. So the Federal Food, Drugs and Cosmetics Act is the basic in the US, whereas in India we go with the ICMR guidelines. And these are for the foods as well as to medicines which the consumers or the patient takes as in form of foods or medicines. So it should be under produced and safe to administer under the good laboratory practices, good manufacturing practices, and good clinical practices, so that it's every part, either it is cosmeceuticals, pharmaceuticals, or nutraceuticals, should comply with the standard guidelines, so that a person, a layman, a common man can take that medicines very uniformly. So, as per the Code of Federal Regulations (CFR), there is a federal register which contains the whole notices and case record format. And certainly, this Section 21 of Federal Regulations contains the pertaining of foods and drugs, and various parts like 21 CFR Part 312, Investigational New Drug Application, 21 CFR Part 314, IND and ND applications for FDA approval to market a new drug. Then Part 316, Orphan Drugs, Part 58, GLP for non-clinical laboratory, also a laboratory animal research, LAR studies, Part 50, Protection of Human Subjects, Part 56, IRBs, Part 201, Drug Labeling, Part 54, Financial Disclosure by the clinical investigators. So these are the various sections of Code of Federal Regulations, like 21 CFR sections, Part 1 to 99, Part 14 is Advisory Committee, Part 50 is Informed Consent, 54 Financial Disclosure by Clinical Investigator, 56 IRBs. Then Part 300, 302 Investigational is IND, Investigational New Drug Application, where the various segments you can see given one by one, like requirement for IND, general principle for IND submission, IND content format, IND safety reporting, annual reports, then the clinical holds for IND and the audits, emergency IND, EIND, or the new drug application, which comes as the NDA. So the content format and the PM, post-marketing surveillance PMS services with the drug, adverse drug experiences, adequate well-controlled studies, then the accelerated approval, orphan drugs, then the BLA, biological license applications, comes up Part 600, and the Part 800 is the devices. So investigational applications comes with the various kind of subsets of rules and the manuals given by this CDERs, and also MAPS, Manual of Policies and Procedures, given by Center of Drug Evaluation and Research, US. So in a nutshell, I will tell that you have a new molecule, its biological activity is done, and you have compliance done with the all processes, experiments, and investigational drug application you have applied. You're going with the medical, chemistry, pharmacology, statistical part. You have done the safety review, and particularly every experimentation part has been done, which comes from phase 0 to phase 1. So routes, dose, and the pharmacokinetic, dynamic, toxicity study, pharmacological approach, toxicological approach, and doses characterization, drug doses forms, etc. have been done, and this reviews goes up, and ultimately when this things goes up, the study starts ongoing. So this is how the IND process goes up. So hope this session will be helpful to all of you. Thank you for joining.